Hi everyone. So since we didn't get to activity 3 um, in lab today, I just wanted to make sure that you guys understood what to do in your lab notebooks before you turn them in before next week's lab. So just to recap what we finished off in lab today. So you guys designed an activity in your breakout groups to test how temperature impacts the enzyme activity in yeast. So in this experiment that you have designed, um, you should have the question, you should have your hypothesis in your lab notebook, including a couple of methods how you tested this question. So how many groups did you use? What was your control? What were your temperatures? Then have a table of your data, which should have been created as you were collecting that data. And then you should have some sort of graph representation of your data, in addition to your conclusion, which should have how does temperature impact enzyme activity in yeast? Could you figure it out? What did your results actually state? Did it agree or disagree with your hypothesis? What were some sources of error in your experiment or confounding variables that you can think of that might skew this data? So that's for the end of activity 2 for your experiment. That's some of the stuff that you should be going over. And then in class, we talked about why different groups addressing the same question might get different results. So this goes along with your sources of error, your confounding variables. So make sure you think about this and you talk about this in your lab notebook. And then just on your own, I would just like you to think about, why do you think that high temperatures might affect enzyme activity? And then also, as an application question, why do you think refrigeration actually slows the growth of microorganisms in food? So this link is to it's a website, and what you do is you double click on the page and it pops up a little message box. And I want you to write your answer to this last question, to why you think refrigeration might slow the growth of your microorganisms in your food. Why do we use refrigeration? Why do we use cold temperatures to slow down growth versus high or medium temperatures? And so this is directly applicable to the experiment we did today, and just something that we actually use in everyday life. So it's important to understand how what we did applies to just general life processes of not only our own biological processes, but other organisms and how they might impact our own lives. So while you're watching this, pause this video right now, take a second, go to this link. This PowerPoint is linked in iLearn, and this link is on that specific page. So go to the link in the PowerPoint and add your thoughts, and I will post a picture of all of your thoughts at the beginning of next week during our pre-lab overview. So the next activity is determining the optimal pH for salivary amylase. So let's review what we learned today about salivary amylase. So we know that it is encoded by the AMY1 gene, and that it's produced in the salivary gland, which means that it's in your saliva in your mouth. This salivary amylase, this enzyme, breaks down starch into complex sugars, and it's one of the first enzymes that are used in the digestive system. So let's do a little bit of an overview on pH. What is pH? What's the pH scale? So the pH is the measure of how acidic or basic something is. The smaller the number, the more acidic it is. So 1 is the most acidic and 14 is the least acidic. We also say 14 is the most basic. And then 7 would be neutral. So 7 is usually where water is. And then also, just a fun fact, your body is a little bit around 7. So our like chemical processes all equal out to a pH of about 7 for our own bodies. So really basic liquids would be like bleach and cleaning solutions. Really acidic things would be like lemon juice, tomato juice. The most acidic at a 1 would be like the fluid in your stomach that breaks down food. So that's actually very acidic. It's at a 1. And that's why we have to have huge layers just a lot of layers of mucus and epithelial cells and other things surrounding our stomach, so that way our stomach doesn't digest itself. So we actually have to have a lot of protection against the gastric acid in our stomach that breaks down food, because it is so acidic, it's actually dangerous to our own bodies. So for this activity, you're going to be determining the optimal pH for amylase. So if you remember, we talked about how environmental factors can affect the way enzymes function. So we looked at temperature as a way that impacts yeast enzymes. So now we're going to look at pH as it affects amylase in your saliva. So in your lab manual under activity 3, there's a table, and that table has pH and then the time it takes for the starch to disappear. So you're going to construct a line graph. Make sure you use the line graph worksheet that's in iLearn. It's the first or second module, to construct a appropriately labeled line graph. Then I want you to look at that line graph and form a conclusion about what you think the optimal pH for amylase is. So this will take a little bit of thinking. It won't be as intuitive as you might think it is, because the data that you have is the time it takes for the starch to disappear, not for the reaction to progress or for the product to appear. So just be aware of that. Let me know if you have questions, and then write what you think based on this data, based on the graph that you got from this data. Write what you think the optimal pH for amylase is to function. So we were going to do a poll based on what's the optimal pH, but I can get together all of the numbers from your lab notebooks and we can talk about it in class next week. And then I want you just to think about this. I would love for you to put your answer to the second question in your lab notebook if you watch this video. What do you think would happen to the amylase in your saliva when it's swallowed into the stomach? Because remember, the pH of the stomach is very low. It's super acidic. So what do you think happens to amylase when it's swallowed into the stomach? And how do you think it would function? So based on what you think the optimal pH is, you can draw a conclusion for the second question. And then this is just something for you to think about as you're submitting your assignments for this week. Just reflect on why you think it's important even for us to understand why enzymes work the way they work, what they are, how they impact us. Why is it something that you need to know about? Why did we spend 3 hours doing this lab? And then these were some of the questions that you guys asked in class today that I didn't know the answers to. So while you guys were spending time in your breakout rooms designing your experiments, I was looking up the answers to your questions. So someone asked about gluten intolerance and if that was the same case as missing enzymes. I found that it's not a case of a missing enzyme, but it's actually an inflammatory immune reaction to eating gluten. So it's similar to an allergy, to where your immune system reacts unfavorably towards something you've ingested, but the symptoms are very different. And then someone asked about the enzymes that influence muscle recovery. So I found this one that's naturally occurring in your body. It's called protease, and it actually is involved in the making of proteins and amino acids, and amino acids make up proteins. And so overall, this enzyme in your body actually aids in muscle recovery and helps ease your soreness. I also came across a lot of health websites and magazines in my very brief search. So I found this one in particular is called bromelain. So this is found in pineapple. And research has shown that it actually aids in the body's response to inflammation. So when you feel sore, it's because your muscles are inflamed because you overworked them. So people take bromelain as a supplement, and there's some research to show that this works. But a lot of the info I found was from health websites instead of peer-reviewed articles. So I'm unsure, I'm a little skeptical about the actual, if it's actually beneficial, but I will say that a lot of the peer-reviewed articles did have cited studies where they said this was a double-blind study, this is how many participants they used, this was the placebo, all that stuff. So they did have cited studies in their articles. I just, I skimmed, I didn't really go into that deep. I didn't have time to go into that deep of the research. I just didn't have time to research it with you guys completely because I was on a time crunch. But there has been some preliminary research that I could find that was done that said that this actually helps your body respond to inflammation. So a lot of athletes will take this as a supplement because they work out a lot and they need to have their body needs to recover quicker so they can keep practicing. So the second part, we still have some more questions from the class today. So someone asked about fructose intolerance and how rare it was. So I found out that it's fairly rare. So only 1 in 20,000 to 30,000 people have it. And it's a genetic condition. And I actually found that there's no real cure. You have to follow a fructose-free diet. And if you don't, then health issues that arise can be as bad as your whole liver failing. So this is a pretty restrictive disease to have. A fructose-free diet it's hard to come by. There's a lot of things that have fructose in them. So this is something that if you have it, you have to make sure that you follow this fructose-free diet because it can really affect your health if you don't. I also found out, I didn't know this, but lactose intolerance, there's a bunch of different types. So there's 3 main types that I found. So, and they're all called lactase deficiency instead of lactose intolerance, but it's basically the same thing. It's you just don't have the enzyme lactase. So the first one is primary lactase deficiency. And this is the most common and starts at the age of 2, but you might not see symptoms till adulthood. But it starts at age of 2 because that's when breastfeeding and bottle feeding has stopped. So once babies don't need to be drinking milk and they get weaned off of it, you might see this lactase deficiency popping up. The other one is secondary lactase deficiency. And this is caused by an issue in your small intestine. And so this is where your body's production of lactase naturally declines. So remember how I was talking about in class where in some cases the older you get, the more intolerant to lactose you are. So that's actually secondary lactose deficiency. So the older you get, the less you're able to really deal with lactose because of issues in your small intestine. And then the last one, this is very rare. You actually have to get the genes both from your mom and from your dad in order to have this condition. But it's called congenital lactase deficiency. And this is where as a baby, you can't have lactate. You can't have lactose. You don't have the enzyme to break that down. And so you have to go on a lactose-free milk formula in order to be able to survive because it can cause some real issues. If you as a babies are supposed to be able to consume milk. That's supposed to be normal. They are supposed to be able to have lactose because they're making lactase in their bodies. But in some rare cases, they can't make lactase. And so this is the congenital lactase deficiency. And there's actually lactose-free formula for children that have this issue. And then we went through the announcements as usual. Don't forget the pre-lab. You have to start it early. I am begging you to look at it this weekend because if you don't, if you wait till the last minute, you're going to be screwed. I'm telling you this right now. It takes a long time to deshell your eggs and to put them into solutions. So start now, especially because there's a chance you could run into problems. You want to make sure you have the time to fix those issues before next week's lab. And I'm here if you have questions. My office hours are Wednesdays at 1. And I'm also available by email. Okay, that's it. Thank you guys so much for listening. I hope this helps kind of alleviate some of your uncertainty around activity 3 so that you can complete it for homework and submit your notebook assignments. Have a good weekend, everyone.